actually, this should be a roundtable discussion, and then I have a coffee break, and thereafter I have again a presentation to go into more detail about showing diverse effects of T-cell measurements in cancer settings. So for this reason, I divided the presentation in a way that they do a general introduction. So once you induce vaccinations and all your treatments in the patients or in your studies, you want to follow up and actually measure them in clinical trials. So what are the challenges when you come to the setting that you finally want to implement it in the system, in the humans, and then want to go in there and measure it? So this is the thought process which I want to present now in this presentation, and which we will discuss together and try to address the challenges you are facing to actually then measure those responses in the human settings. So as I'm from a company, I always need to make a first slide where I show where I'm coming from. I'm an adjunct associate professor from Case Western Reserve University, founding cellular technology, and I am a co-founder of this company, which has 3 segments: the contract laboratory, which I'm representing to help you implement the measurement, and then the consumables for standardization processes and the instrumentation to allow you to measure it yourself. With that one, I will not talk about the company anymore, but I need to do this to show that. Okay, so what are our rationales of monitoring cell-mediated immunity during clinical trials? All of you are very aware of it, that cell-mediated immunity is a critical component during most immunological responses involved in all types of diseases: cancers, autoimmunity, infectious diseases, transplantation, allergies, and others. The efficacy of therapeutic vaccines and other immune-based interventions will depend on the de novo induction of modulation of existing cell-mediated immune monitoring. The basic research which we do and did showed us very clearly that correlates of protection has exposed the limitation of the vaccine approaches that rely solely on antibody responses to confer protection against pathogens. We need to look and define the frequency of antigen-specific T cells is one of the primary goals of ex vivo T-cell diagnostics. However, for this reason, what we found and saw is that non-classical PK/PA mechanisms of actions apply. The drug is not biologically administered, but it is a therapeutic vaccine, meaning that moiety is the resulting in vivo developed altered specific effector T cell and B cell response, which then induces the control or the effect of your vaccine treatment. Responses characteristics, they are slow, require repeated dosing, and patients with malignancy often initially progress before an objective response is detectable. So then when we want to measure the implementation of these responses in cell-mediated, then we need functional correlative biomarker assays that require living sample and cell material to measure this. This cell material needs to be sufficient in quality and quantity to be collected in the clinical size, and most of the times it cannot be directly tested by the clinical sites themselves. So you will need laboratories which can handle living materials. These assay systems need to be effective, need to be validatable, standardizable, and needs to be primary cell-based assays, not cell line-based assays to closely represent what happens in the human system. It should measure physiological and clinical relevant responses and should have adequate sensitivity, meaning if you look at T-cell responses, you cannot measure antibodies, etc. So you need to really find the correct correlation They need to be validatable, they need to be accurate, high throughput capable, and you need to have adequate systems in place so that you can follow and measure them in a CFAR Part 11 compliant manner. Further, to implement it, as you work with the human systems and you will have many samples, they should perform identical with fresh as well as previously frozen materials so that you can standardize and batch the sample measurements together. They should work with an outbred population, genetically diverse, because otherwise you reduce your vaccine approach to a very small population and give all the challenges with that. So, and as you work with humans, you want to use as little cells as possible, yes, to draw blood, because you need to collect the samples accordingly. It should be scalable, so it's not something which you can do in the lab one by one, that's okay, but once you are in the clinical setting, it needs to be done in higher numbers, so you need to be able to scale it accordingly and measure it accordingly. So what type of measurements are currently used in the setting by most of you, of the people? And all of you are familiar with the classical enzyme-linked immunosorbent ELISA assays or the Luminex and cytokines or multiplex bead arrays. However, you need to remember all these assays are supernatant based, meaning the released product is already released. It's not produced anymore. So there are different consequences of that and frequency and sensitivity measurements of that. So it doesn't matter how many cytokines or how many products you measure, it will always apply to a certain sensitivity. The multiplicity of analytes measured will not increase the sensitivity of the system. It just provides you more readouts to multiple analytes or cytokines, as they are all supernatant based. So the samples and the products are already diluted, absorbed, produced, and has all the consequences with it. Then you have very few assays which are really cell-based assays, yes, where you require materials where you have cells there, like the enzyme-linked immunospot assay, the ELISpot assay, or flow cytometer-based measurements. Each of those assays have their limitation and their sensitivity. Now, you need to know that when you apply them. What does it mean that each assay has a different sensitivity, even though they measure the same product, the same analyte? Yes. So for this one, I draw here. Okay. Then I use the mouse. Yeah. Here I showed the classical 3 type of assays which you could use. And here the idea was to show you what does the different sensitivity mean? What is the consequence of it? All of the 3 assays measure interferon gamma. This is an example where we now used, for simplicity, to explain what it means. We take, say, CHO cells which are transfected to produce and release interferon gamma, mix them with CHO cells which are not secreting the cytokine. Then we titrate in the number of transfected cells, and then we measure how many cells do you need to secrete interferon gamma to detect it with the corresponding assay system. Okay? That you can do with cell lines, T cell lines, primary cells, etc. So here, so then you titrate in the transfected cells and you measure it, how many you can detect. Here by intracellular cytokine staining, here by collecting the supernatant, measuring that by ELISA. Below that, here with direct ELISpot measurements. As you titrate in the cells, you get a linear correlation by flow cytometry to about 1,000 cells in a million. So you need 1,000 interferon gamma producing cells before you can, until you come to this background noise. Maybe if your system is super good and you collect many 100,000 gated events where you need milliliters of whole blood and sample stain to be able to do that, and do the acquisition for more than half an hour per sample, then you might go down to 500 cells per a million producing interferon gamma cells, okay? So that is your limit. If you have frequencies directly in vivo in the patients or humans which are below 500 producing interferon gamma cells out of 1 million, then you will not see it. It will be negative, right? So that is here. If we do that with ELISA, collecting, placing the cells, doing the culture, collecting the supernatant, thereafter then go and do the measurements in an interferon gamma specific ELISA system, or cytokine B arrays, multiplex assays, all which are supernatant based, okay? You have again a typical curve. You come down to about 1,000 cells in a million producing cells. Then you get an OD of 0.1, barely above detection limit. So again, you need 500 cells in a million to be able to detect it by that system. That's the detection sensitivity limit. If you do it by ELISpot, you have here, if it's over that, you have too numerous to count. Yes, it is plateauing out. And if you go into the lower range, you detect them. It goes 1 in a million, okay? So which means is you have different assays to choose from. And you need to know what is the frequency to be expected in the natural occurrences to select the right assay system. Otherwise, if you choose this system or that, you need high frequencies. This system is good for low frequencies. So, and if you choose this system for low frequency measurements or this system for low frequency measurements, there will be false negative. So your readouts will depend on the right assay system to choose. And this is something you will need to be aware of when you select your readout systems in the primary clinical settings. Furthermore, this slide very nicely illustrates you the diverse sensitivity, which we very detailedly discussed now. It is directly ex vivo measurements, so no cell lines, no cultures, no nothing. It's the same assay performed with also the same specificity is there by diverse sensitivities. ELISpot is for one which do not require HLA typing of individuals. It's applicable to all systems. And the samples are not pharmacologically treated. We talked about that samples need to be able to be tested frozen, as for economic and for logistic sample, meaning if it's not frozen, you would need to test it at the time it is collected. So you have different measurements at different time points, so you cannot really compare baseline measurements versus treated measurements at a later time point if you cannot do the sample measurements with frozen cells. So here I show you the example that fresh and frozen cells behave absolutely the same, measuring antigen-specific responses to interferon gamma, IL-2, IL-4, and IL-5. Measurements, here this is the frequency of the responses measured, so the numbers of spots in the samples plated. Dark are the frozen, light one are the fresh, and they're plotted against each other. The cells have been titrated, so you have a titration curve showing here, and the counted cells per well are dotted. Then we plotted the curve, which turns out to be linear, showing how the efficiency is, and you see clearly that every single spot is counted, and at any titration points, the same frequency of responses is measured, which is one of the validation criteria. It's linear, so you have a linear correlation. then it is not just important that frozen cells, the same numbers are measured, but as well the same quality. And here we plotted then the sizes of the spots, comparing the fresh interferon gamma or IL-2 or IL-4, IL-5 responses to the frozen one, and compared the spot sizes. And you see that they're completely overlapping. So it is valid here, it performs linear, and it has a single cell resolution. And to achieve this, what is another important thing, if you have international studies, and you have some in Europe, some in Africa, some in the USA, Canada, etc., you need to harmonize the sample collection at the clinical site. All of them needs to use the same materials, same tubes. They need to know how to handle it, because living materials need to be handled differently than dead material. And serum is dead material. So 99% of all clinical sites are used to work with dead material. They handle it accordingly, yes? And if you want to collect living things, you need to harmonize the sample collection in all those locations to understand that the clinical site know how to handle the whole blood. Accordingly, proper sample handling, processing, cryopreservation, shipping procedures need to be implemented, SOPs generated. Then you need to harmonize all those across all participating laboratories, implement the details of the SOPs, including the cell thawing, cell counting, assay procedures, and standardized assay materials should be used. That this can be performed, and it's not something which is not possible. Many people say, oh, we don't want to touch cellular immune responses, that is just a nightmare, it will never work, etc. That is not correct. Once all the processes have been implemented, it is very straightforward and very doable. And for this reason, I show you this study, which was performed by multiple sites at multiple locations throughout and different laboratories. And what the results were, we had provided the cryopreserved samples, we provided the wash media solutions and testing. We allowed one variable, which is that everybody, they were all professional cell-mediated, you know how to do responses in cell-mediated immunity. They were allowed to use their favorite tested, superly qualified serum-containing media. That was their one variable which was allowed. We provided the peptides, the antigens, the interferon gamma kit, and the procedures and the SOPs. So as discussed, standardized processes, and we generated, the CTL served as reference laboratory, so we generated what the expected readouts would be and should be, and then provided the labs. The laboratories performed the testing, and these results are plotted here. 3 standard deviation is shown here from all the laboratories. High responders, only this one was a little bit lower than 3 standard deviation to the recall responses measured, but everybody got that in the high frequency, in the medium frequency range, and in the low frequency range. So everybody could reproduce, get the same results, independent where the location was, with the same cell materials and same SOPs. So it was 100% of the labs detected the responses in the same quality, high, medium, low. And of the 9 labs resulted within 3 standard deviation of the reference values. This is again a validation criteria, classical validation criteria, that it's reproducible between different locations in the lab, in different laboratories. The only variable which was allowed was the serum tested their biological super qualified media. And this is the result of that is shown here in the gray bars. So this is the serum containing media, and the other one is the serum free media. And down here is the spontaneously induced immune responses of the cells, and up here the antigen-specific induced responses. This divides each laboratories. This is more or less raw data. 1, 2, you see all the labs. The high, medium, or low respond, high, medium, low responding cells. And what you see here, that in about 1/3 of all the labs, the tested serum-containing supermedia actually was mitogenic to these primary cells in some cases, and the medium responses were high, okay, like in this lab. And here it is 1 out of the 3 human samples, 1 out of the 3 human samples, 1 out of the 3 human samples. In some cases it worked very, very nicely, okay? So it is not serum-containing solution can work. However, in an outbred population, you see about a loss of responses because 1/3 of the human population will respond to the serum and induce a high spontaneous medium response to the serum, which means is even though the antigen-specific responses are the same, comparing the serum-free versus the serum here, you see it's beautifully, it's very comparable. However, as the medium response is high, you lost the responses. So this sample got negative now because you have no difference between the antigen-specific response and the medium response because of the mitogenicity of the serum you used. And the serum exposure can be at any point, wash solutions anywhere. Once the mitogenic response is induced, it's there. You lost that sample, okay? And then what you then further see that in some scenarios, even though it was not mitogenic in media, it was toxic to the cells. So it reduced the antigen-specific responses. So serum is one of the biggest challenges you have to face when you work with cellular immune responses in a broad human population, as even though you tested and qualified it for a given population, it will not be the same for all. So you will always, you can account about 1/3 of your samples be lost because they were exposed to serum at one time point or another. So for this reason, we highly recommend that serum-free solutions are used. And with that one, I would like to finish my presentation and go to the discussion points, because this should be a roundtable discussion, and ask you, what challenges have you faced when you were looking at immune responses in your clinical trials, and how could we, or how could that be addressed and discussed? And with that one, I would like to go over to you. Yes?
Can I go backwards? Talk about standardization. I got confused there, because I still don't see how it's a standardized assay.
This one is a very, so you don't understand why it's standardized. Yeah. Okay. So it is standardized because everybody got the same procedure. They used the same protocol, the same cells. Everybody got the same cells, yes? There's the same collection time points from 3 different human samples, where we have pre-qualified those samples and pre-tested them in a reference laboratory, which were ours, where we defined the acceptance value, what is an expected results to an antigen of a given antigen, and provided the cell materials through
are they a high,
different,
medium, low?
Excuse me?
Are they a high, a medium, a low?
Correct. Okay. Responders. So we had high responders, medium responders, and low responders. Right. We had pre-screened and selected those samples. Of course, they're the best responding, otherwise, you know, and we knew the titration risk, just to make sure if you have the same protocol, the same cells from the same collection time points, provided them to diverse labs, and we shipped out all the materials to all these laboratories. We provided the wash solution, the test solution, everything, yes, including The antigen, which this antigen was a CMV peptide 65, here's the sequence of the peptide which was provided. We provided the kit, the ELISpot kit which was used, so the assay readout, and the protocol. The only variable was that they were allowed to use, beside all the given materials, their serum containing solutions.
Okay, so the question then is, did all the labs swap their favourite serum?
Yes.
And is that's where you had the
That is
variability, right?
this one, yes. That is this is these grey bars. Correct.
Okay, so the standards work then, the medium.
The black ones.
The standards work high, medium, low. But
Correct.
The favourite
High,
serum
medium,
wouldn't
low.
work.
Yes, and the favorite serum changed that behavior.
Okay.
Yeah, because suddenly the high, medium, in some cases it performed very nicely. I don't say it does not it cannot perform. Yes, it can. However, you have, as you work in a human population which is outbred, the variability will always be there. And as you pre-screened, it works to a certain extent, but you will lose about 1/3 of your in the human population. About 1/3 of the humans, even if you prescribe, will respond to the serum, because serum is a very crude protein mixture with all types of products, and 1/3 will be mitogenic, so it will induce a spontaneous interferon gamma response, or it will be toxic, and it will actually reduce the antigen-specific response. Okay? Yes?
You know how the serum labs selected their own serum, which is the test to select a good
The
serum?
serum, usually when it is done, is a very classical way how you do that. You get about 30 different batches from 3 different vendors, okay? Then you screen, and you have primary cells or hybridomas. First of all, you need to heat inactivate it for 30 minutes in water bath, cool it down, filter it, get away all the proteins. Then you test it in your assays, and out of the 20, 2 will qualify, meaning that they are non-toxic, non-mitogenic, to the selected cells of your choice, what you have. Once you did that, then you buy a giant batch and spend $20,000 or more, and put them into freezers, aliquoted, and they're good only for about 1 to 2 years. Yes, so if you run over that, you need to requalify your original batches. So you store them, your favorite super tested serum containing solutions, serum which you then use in freezers, and then take out aliquots. Each aliquot needs to be heat inactivated, filtered, and so on, and then used in the assay, and you use it constantly. So the same lot of serum will be used through 2 years, yes? And then about 2 years it expires, and you can start all over again. Yes? So it's a constant challenge. And sometimes also 20 lots you test from the different vendors, and that's quite a work, because think about it. You need to then take all these 20 lots, all of them you need to separately heat inactivate, filter, do it, have 20 medias in the same assay, screen all the cells. So it's a major effort to do the first step of screening. But once you did it, then you have qualified the serum media. But even if you did that, in a human population, as it's outbred, you still will have this 30%. If you do not do that, the pre-qualification, then it will be much higher than 30%. Then it might be 100% of the cells responding with toxic or mitogenic responses. Because that's why how serum disqualifies, because it just kills the cells, yeah? So this is one of a very critical cellular immune response prerequisite.
Yes, because from my experience, when we use serum, we check on the T cells, T cell proliferation, on spontaneous proliferation, but also on differentiation of the dendritic cells.
Yes.
And they are very sensitive to endotoxins or other So if you don't get a good differentiation of the dendritic cells, then you don't get a good serum. That is our best selection cell lab to test the real high quality serum. We get, because if you have any, the phenotype of the dendritic cells at the end of differentiation will be harsher. They will not express it as some kind of markers. That is one of our experience.
Correct. So this is what, in my experience, was the biggest challenge which everybody faced and most people were not aware of. And most laboratories don't care. They provide the frozen cells, and you don't ask them, so how do you do the freezing? Yes, and then they wash with serum, they freeze serum free, but do the dilutions and preparation of the PDMCs in serum containing solutions, freeze serum free, and then doesn't matter, it has the same effect already in the process. So this is one of the critical points I found which needs to be addressed during the collection of cell-mediated material for your future readouts. Because if you have inappropriately prepared samples, whatever and how beautiful your assay will be, it will not work. Yes, and then you spend a lot of money on the clinical trial, and you have a lot of challenges to face with. So that was, yes. So what other challenges have you faced?
So can I go back one step again? So
Yes.
Sorry to be a pain. But it seems to me ultimately you're looking for a corollary protection.
Yes.
And ultimately that's what regulators want to see.
Correct. That was this point where we said here in the discussion that you need to select the assay of your choice, right? That it measures the and clinical relevant responses.
So have regulators given any opinion on what they feel about these sort of assays?
Yes, until you developed it and optimized it to your antigen-specific responses, yes, then that will be the assay of your choice. They don't say what you need to use. They give the choice to you, but you need to optimize the assay. And
But you've still got to prove to a regulator that it's reproducible and you're going to have a standard that you can use.
Correct.
And everybody loves antibodies because you can make international units, you can store your anti-sera forever, and
yeah, but
reference labs.
that is true. But for any assay you use, all the standards are never forever. So you need to.
Okay, but you can bridge with antibody assays much easier than you can bridge T-cell assays.
Yes, but antibodies, and that will be my next presentation, do not measure T-cell responses. That's like looking out to B cells here, and you want to look this. So then you look at the shadow, and you hope the sun falls correctly, and the B cell response, which is an antibody, an immune globulin response, will reflect the T-cell responses. And that might or might not be the case. You might have luck, or you don't. So it needs to be relevant. So if you want a T-cell assay, a measurement of T-cell responses, you should not look at antibodies.
So you misunderstood me. What I was actually going to say was, when you look for a correlate, you don't mind what it is. It could be anything.
Yeah, that's true.
And antibodies, as you were saying, are easy to do.
If you have shown that the correlate...
If it's appropriate, I agree.
Correct. And that's a chance question.
Of course. But I can't think of any vaccine that has a standardized, validated T-cell assay for correlate protection.
There are.
Maybe TB.
There are. We developed and did some, and you can validate them, and that's what I showed you. And that's why I want to show and make you feel comfortable. If you do a proper preparatory work, yes, cellular T-cell response measurement are validatable, high throughput capable, standardizable, everything you want. But you need to properly set them up. And it needs to be differently set up than with antibodies. And if you don't do it, then it will not work. And because dead material is handled differently than living materials. Okay, it starts with shipping, Some people use room temperature shippers, okay? They put the sample in the cardboard box, okay? Then ship it. Fine, everything is shipped over the airport. The cardboard box doesn't protect from light. So now you sit it out on the driveway, sun shines, we have 30 degrees Celsius outside in the cargo where it stands for 2 hours, it gets 60 degrees, you boil the sample. Of course the cells are dead, yes. Everything will be dead thereafter. So, and antibodies are much, because they are not living samples. They already produce proteins. Their survival is higher, so you have a higher chance it works if it's inappropriately handled. But not with living materials. You need to, I call them controlled room temperature shippers. Okay, it's not a cardboard box. It's isolated, it's protected, it's temperature monitored, it's controlled. Yes, to maintain. You don't, livable, because now you work with living material and different handling are required. And that's my goal. I just want to, that's one thing you need to look about. Yes, you're right. Okay, going back to the questions. Yes.
We have tried this human clinical trial, phase 1, and we completed a collection of the samples, especially we did collect the PBMCs from these subjects. And each site, the clinical trial sites, they do have different personnels, they do have different characters.
Correct.
So it is very difficult to standardize unless you really go and supervise them how to preserve the samples before you then ship it out.
Okay.
And so I think there should be some sort of standard technique that you, as maybe the CMO, just tell them how to preserve the samples, how to ship it, and what samples are acceptable and what samples are not acceptable, and so on and so forth. And we had 4 sites, 2 in California and 2 in Washington states. And when we collected those PBMCs, we did have trouble getting those good samples out of those people.
Yes.
So how would you suggest that they should be standardized, and then we can collecting agent or collecting place, we have to then ship it to the CMOs. And now I assume that the CTL, and that's a very nice company name, CTL.
Yes.
And do you provide any kind of advice for those people who are doing the clinical trials?
Yes, we do. Yes. We have worked on that for over 15 years now, how to standardize it and implement it. Yes. And we generated, I call them temperature controlled room temperature shippers. Okay, so boxes where proper sample is preserved and properly handled with temperature monitors, and we actually do training of the clinical sites, because as they're used, 90% of what they do is they draw blood, put everything in the refrigerator. That's what they learned. With serum and antibodies, that's what they do. However, we know that if you put cellular materials into the refrigerator, you lose immediately 30% of functionality. Right there, right done. They have done everything perfect, just stuck it in the refrigerator. So we actually do trainings of the clinical site from the phlebotomist that once you draw the blood, properly label it, you are not allowed to put the samples into the refrigerator. That's starting point 1. Okay? Yes, and then provide the proper boxes for the shipment of the samples so that room temperature is maintained. Sadly, most clinical sites do not have proper equipment and training to separate PBMCs, meaning they don't know how to work in BSL-2, they do not have hoods, they don't have -80s, and they do not have liquid nitrogen tanks. You cannot store PBMCs, cryopreserved PBMCs, in -80 Celsius freezers or -150 Celsius freezers. It needs to be stored in liquid or vapor nitrogen tanks. If you don't do that, the functionalities of the cells after 3 weeks go down like nothing, and you lost all your samples. So my recommendation is, and that's why I said, of course, as we do it, I have a conflict, and I clearly stated that at the beginning, and but that's what my job and my work is. We do this and we standardize this and we can do it, and that's what I showed, yes.
Yeah, some of the samples are HIV positive.
Correct.
So how do you
That's
handle
not a
those?
problem. Every human material is biohazardous, yes. And as you work with primary cells, HIV samples, until you don't culture them for more than 10 days, you can use them in classical BSL-2 hoods and work accordingly. That's not a problem. The same with hepatitis C, hepatitis B, etc. All human materials are handled the same way. It's a classical BSL-2 handling, yes. Any other questions or suggestions? What challenges you have faced in your clinical trials? Because we can, yes.
I just want to tell you that we've completed, I think, 5 now clinical trials using CTL media.
Yes.
We transported, everything in green boxes, the temperature control boxes. We've transported frozen cryovials and liquid nitrogen through cryopores. And it's really not that difficult to set up. Once you have the right protocol, you go out and you train the CROs. And right now it's difficult because T cells are a new thing. T cells and vaccines are new and people don't know how to do it, but you can go out and train these people. And we've had success doing that ourselves. We actually worked out all the conditions ourselves in the lab where we had blood transported to us and then we processed the blood. So we knew what was going to work and what wasn't going to work, and we've taken it from there, and it's worked beautifully. But it really is important to go out and train the CROs that are taking the blood. And you often have to supply them with things like liquid nitrogen containers or -80 freezers. And it sounds expensive, but when you think about the cost of a clinical trial, it's not, because those samples are so important. If they're not frozen and taken correctly, then you've lost everything. So it's not that difficult, and I feel very strongly about CTL reagents. They've been fabulous. Serum-free reagents are the way to go for sure.
We're lucky that our samples don't need to be preserved because we're looking at barcode and as well as copies in PDMCs, and we sequence them all. And so that doesn't require
But if you wanted to
live
look,
cells.
right, if you wanted to look at T cell responses
Yeah.
in your clinical trial of your vaccine, you will have
Yes,
to
then
try and preserve
you
the
have to
sample.
preserve, yes.
Okay, because this is something you need to think about when you set up your trials, because again, if you have wrong material from the beginning, you spend millions and then you lose it because of small things. And that is what is so sad about. They are very tiny, small things which will implement and make the chat processes. So then my next question is, what standardized cellular readouts have you used? Yes, so as you said, you did PCR, you said you worked with other assays. So as you said, that you don't really believe that cellular standardized assays can be used. So what are
I'm not sure I go that far. I would say I have reservations. Like most people, my experience on the antibody side is a lot easier.
That's true.
The T cells have a lot of challenges. But as Wendy was saying, if you have SOPs in place, then it's achievable. I think the question I had was about standardization between different sites and trying to compare the data.
Correct.
And I think you addressed that very well.
Right, I try to. That's what my point is, and I'm a strong believer in T-cell responses. That's what my scientific career is, yes, and that's what all my publications are. And I just want to say it is doable, and don't let yourself discouraged, yes, and it's not so difficult. I hear so many negative things, and I'm not happy about that because they sound to make it so complicated, and it's not, yes. Of course, I can go over and list all the 1,000 things which can go wrong, but in any assay, anything can go wrong, and you need to look in a positive way how you can address it and make it workable with very little but it needs to be preempt what you know, what the challenges are, address it ahead of time so that you don't get the big surprises at the end. That's my goal, what I want to achieve and discuss here, and encourage you to really not look at the shadows and hope they correlate, but go and try, at least try to look at the correct readout systems. If you want to look at T cells, look at T cell responses and not B cell responses, yeah? As they're different, and all of them have valid readouts and valid, but it has implications. And I will discuss that in my next presentation after the coffee break. So this was my second question. Then the third question is, again, are always viable cells needed? Of course, if you look at flow cytometry cell surface markers, no, yes? And that was my big other surprise. Oh, we do all the time blood processing of PBMCs. We use validated FDA-approved tubes. I said, great. So what is it? Yeah, and it says validated for flow cytometry, maintains whole blood for 3 weeks, measurable for cell surface markers. I said, great. That's good for cell surface markers. But I don't believe it's good for viable functionality measurements, because how can cell surface marker be stable for 3 weeks only if the cells are actually preserved, okay? If they are fixed, and if they are fixed, they don't function anymore. So you cannot collect the same samples from with the same collection tube, fixed cells for first surface markers, and then measure functionality. So these are some other issues. So you need to know what you're using the cells for and what the readout is, yes? Any other comments? Okay.
I just want to say something that is, because we use, I'm a T-cell work mainly, but it is, I agree with you always, but I think it's very important for a vaccine, because you need to both be in values. So we can be, because you cannot take blood of people like, so you have the amount of blood to take is in some way limited. So I think it's very important to know exactly what we want to see, what is important to see, and try to collect the minimum samples for separately from antibodies and T cells. I think it's very important.
Yeah. There I want to give you another thinking process, limitation of materials. Why are people not using plasma? Yes, why is always serum needed? Okay, plasma, if you collect 30 milliliters of blood and you collect the interface for white blood cells, PBMCs, you have 20 milliliters of 1 to 2 diluted plasma with PBS, and you throw it out. And then you collect 5 milliliters of whole blood and collect 5 milliliters of serum. So if blood is so limited, why can plasma not used for antibody measurements? Okay. I don't understand that. So this is a question I have.
It depends on the anticoagulant that you use. You use heparin or another thing, it could interfere in your test.
Correct. But can that be tested ahead of time? Most of the time when I ask the people why they don't use plasma, they said they don't feel comfortable with it because they develop the acid in serum and they don't know what the interference of heparin is. So my question to you, is it not worthwhile to test that ahead of time and see if this would be something where you could use, how the effect of the anticoagulants is on your ELISA systems, because then you could actually use the materials, from different points, and this from the same collection have serum, have plasma, you have much more material with much less blood. So that's another thought process which I would like to point out too. My last point of discussion is what collection method, and actually we started now going there, that the different anticoagulants are as well very important to look at, yes? And there as well, it matters if you use natrium heparin, lithium heparin, ADTA, citrate, what else have I seen? Many other anticoagulants, they have effects. And yes, you are 100% correct. It needs to be properly addressed before you collect and go into the clinical trials to see what you need to do. In my experience, natrium heparin, classical natrium heparin anticoagulants works the best for PBMC collection, and it allows you to ship samples for 24 hours overnight. It works perfectly fine if properly handled. The interferon gamma responses are maintained and similar and equal. That's what the data was shown at. And if the samples are shipped for 48 hours, interferon gamma still works. It's about 50% lower, okay, but still gives you responses. And after 72 hours, no responses, then the sample is useless, okay? But 24 hours is absolutely no problem if properly handled and correct blood, and that's important because that saves you all the trouble, because there as well, there is a lot of talk out that it needs to be just immediately processed, etc. In our hand, no. 24 hours is equal. 48 hours after blood draw is reducing. I don't say it's the same anymore. It's reducing the responses, but still antigen-specific interferon gamma responses are very good measurable after 48 hours. However, they're about half of what they would have been by 24 hours.
So what's the comparison with EDTA?
Excuse me?
What's the comparison with EDTA? So if you just ship the blood of EDTA, what's
EDTA is not as good.
not
Yes.
so good?
Each anticoagulants will change the kinetics of the times, how it is. So for me, yes, it is doable. 24 hours is not so critical, but then if you go at longer time period, the impact of the anticoagulants will be more and more pronounced. That is definitely the case.
You're talking about function as well as...
I talk always about function. Yes, I always talk about
What
function.
about the viability?
Viability is misleading. Viability does not necessarily correlate with functionality. So what we have seen is that, I don't know how and what they did, because you fiber the samples, so they appear to be viable. Yes, so you get beautiful viability data and zero functionality data. So when we qualify sites, we central laboratories, we always measure functional assays. So, and I don't really, okay, viability once we thaw the cells, then the viability is a much better correlate than before freezing. So because you cycle, then you always have high viability. But once you froze the cells and you thaw them, the viability should be above 50% to get good data. You can see only problems if the viability is below 25%. Then even nitrogen doesn't respond anymore. So,
Magdalena, I think
yes.
we should take about
Break, and we
15-minute
can
break,
discuss.
and then you're going to talk
Right.
afterwards. So let's take a 15-minute break and then come back, and she's going to give us the first
Right.
talk. Okay.
Okay. Thank you for participating in this round
Well,
discussion.
that was very wonderful, and it's a good experience to share. Yeah. Right. Okay, so let's come back.